Imagine just for a second that you are hovering in a helicopter looking down at this satellite map of a massive, sprawling city.
Ooh, I like this.
Right. And if you pull out binoculars and just start counting, like every single brick, every pane of glass, every patch of concrete, you might feel like you're gathering a ton of data.
Yeah, but you actually know absolutely nothing about how that city works.
Exactly. But if you zoom out a bit and, map out the actual neighborhoods, if you could say, okay, here's the financial district. Over there are the quiet residential suburbs, and down by the river are the industrial zones. Suddenly, you actually understand the city's heartbeat.
You see how the money flows, where the traffic bottlenecks, all of that.
Right, how it functions as a living system. So welcome to this deep dive where we are going to do exactly that. But instead of a city, we're mapping the human body.
It's really a perfect analogy to start us off, because today we are exploring this just incredibly groundbreaking paper that was recently published in the journal Nature Methods.
Yeah, and it's a dense one, but so fascinating.
It really is. It introduces this highly sophisticated AI algorithm called CytoCommunity.
CytoCommunity, got it.
Yeah, and what CytoCommunity does is map what biologists call tissue cellular neighborhoods, or TCNs for short.
TCNs, okay.
Right. It basically looks at how our cells physically organize themselves into these specific communities to support, healthy tissue function, or critically, in the case of cancer, how they organize to build a fortress and actually drive the disease.
Wow. So our mission today, for everyone listening, is to leave absolutely no stone unturned. We're going to unpack the mechanics, the AI architecture, and the real-world biological case studies inside this CytoCommunity paper.
We're going deep.
We really are.
Yeah.
We want you to understand exactly how this AI thinks, why it's beating older technologies, and why mapping these cellular neighborhoods is just this massive leap forward for the future of personalized medicine. So let's just jump right in.
Sounds good.
Before we get into the AI itself, I feel like we need to understand the biological puzzle it's actually trying to solve, right? Like, why couldn't older methods just draw these neighborhood maps?
Yeah, so to understand why the older methods failed, we really have to look at how they perceived the tissue in the first place. A piece of human tissue is not a blender smoothie of random cells.
Right, it's not just mush.
Exactly. It is a giotto structured, organized community. And there have been previous spatial mapping tools that tried to find these communities, tools like Giotto, BaseSpace, or, spatial LDA, UTAG.
Just like a whole alphabet soup of algorithms.
It really is. There's SpaceGCN, STAG8, SpaceGM, but they all struggled fundamentally because of the raw data they used as their foundation.
Okay, what kind of data were they using?
Well, many of them looked at raw gene expression data.
Wait, isn't looking at the genes a good thing? I mean, we're always taught that DNA and genes hold the ultimate blueprint, right? Why would looking at gene expression make a mapping tool fail?
I mean, in theory, it sounds great, but practically, raw gene expression data is incredibly noisy.
Noisy how?
Well, if you're looking at thousands of genes per tiny spot on a tissue slide, that expression just fluctuates wildly. It's too chaotic to draw clean borders.
Oh, I see.
Yeah. And on the flip side, some older tools tried to use spatial proteomics, so looking at the actual proteins instead of the genes.
That seems like it would make more sense.
It does, but you only have maybe a few tens of proteins to look at, so the data is incredibly sparse. It's basically either a deafening noise with the genes or a quiet whisper with the proteins.
Okay, so the data itself was either overwhelming or totally underwhelming. But didn't some of the newer AI tools try to, fix that issue?
They did. They tried to fix it by using what we call intermediate clustering.
Intermediate clustering.
Yeah. So they would use a neural network to turn the noisy cells into a mathematical representation, basically just a bunch of numbers.
Okay.
And then they'd run a completely separate secondary clustering algorithm on top of those numbers just to group things together.
So it's like a two-step process. Why is that a bad thing?
Because it disconnects the actual real biological identity of the cell from the final map. The borders they draw become totally dependent on mathematical quirks of that secondary step rather than, the biological reality of the tissue.
Ah, I see. So to go back to our city analogy, it's kind of like trying to figure out if a building is a bakery or a bank just by looking at the raw fluctuations in its electricity usage, rather than just like walking up to the front door and reading the zoning label on the glass.
That is exactly it. You're guessing based on secondary signals.
Aye.
And CytoCommunity completely changes the game because it bypasses that noisy raw data.
How so?
Instead, it uses direct cell phenotypes as its foundational data.
Phenotypes meaning like the actual final identity of the cell.
Yes, the pre-annotated cell types. It already knows, okay, this is a T cell, this is a macrophage, this is an endothelial cell.
Oh, wow.
Right. It takes those established identities and learns a mapping directly from the cell type to the neighborhood space. It just completely skips the middleman.
Okay, let me make sure I'm locking this all in, and I want you listening to visualize this too. So to recap this first part, tissues are structured into functional communities, which we call TCNs.
Yep.
And the old mapping tools were getting lost because they were trying to draw borders based on noisy gene static or like clunky two-step math that just lost the plot.
Exactly.
And CytoCommunity fixes this by looking directly at the actual identities of the cells to figure out the zoning laws.
You got it.
So think about your own body right now as you're listening. Think about your immune system. Imagine how a neighborhood of immune cells might look entirely different spatially when they were just calmly patrolling around a healthy cell compared to how they physically swarm and organize when they surround a cancerous tumor cell.
Right. That physical arrangement is literally everything.
It really is. And the natural question is, how does an AI actually draw the borders of those invisible neighborhoods without a human pointing and clicking on a screen?
And this is where we dive into the mathematical engine. CytoCommunity uses a graph neural network.
Okay, I've heard of regular neural networks like, the ones that recognize faces in photos on your phone. What makes a graph neural network different?
Well, a regular neural network is great at looking at grids, like the pixels in an image, but cells in a tissue aren't arranged in a perfect grid.
Right, they're kind of messy.
Exactly, they're scattered organically. So a graph neural network is designed to look at relationships and connections kind of like a social network. So step one for CytoCommunity is building that network.
Okay.
It builds a k-nearest neighbor graph, or KNN, and this is based purely on the physical Euclidean distance between the cells on the slide.
Okay, my brain really needs a visual for this.
Mm-hmm.
Is it kind of like casting a microscopic spider web over the tissue, where every cell is a dewdrop, and the AI connects a silk thread from each cell only to its immediate closest neighbors?
That is a brilliant way to visualize it. Yes, a very precise spider web. And the algorithm actually calculates exactly how many threads each cell should cast out. That's the K in KNN.
How does it know what number to use for K?
By default, it's set to the square root of the average number of cells in the sample.
Oh, clever. Okay.
Yeah. So once the web is woven, step two is giving every single cell on that web an ID card. It uses a mathematical technique called one-hot encoding.
One-hot encoding. Break that down for us. How do you turn a biological cell into an ID card?
It's just a way to turn categories into clean math. So let's imagine our tissue only has 3 types of cells: B cells, T cells, and macrophages.
Okay, give me the simple.
Right. The AI gives a B cell a vector of 1, 0, 0, a T cell gets 0, 1, 0, and a macrophage gets 0, 0, 1.
Gotcha.
So it's a binary code where only the specific cell type is hot, meaning it has a 1 and everything else is switched off to 0.
Clean and unambiguous. I like that.
Yeah.
So we have our spider web, and every cell is wearing a very clear numerical barcode. What happens to the web next?
Now it enters the actual graph neural network. The graph goes through a convolution layer.
Okay.
And as it processes all those connections, it uses a noise filter and activation function called ReLU.
ReLU. What does that actually do?
Rectified linear unit. Essentially, ReLU stops the math from spiraling out of control by turning any negative numbers to 0, so it only lets positive signals pass through.
Oh, that makes sense.
Yeah. And the output of this layer is a cell node embedding matrix. It's a high-dimensional space, typically set to 128 or 512 dimensions.
Whoa, 128 dimensions.
Yeah, AI loves high dimensions. It gives it a highly complex mathematical understanding of each cell and its immediate neighbors.
But wait, we still don't have our neighborhoods right. We just have incredibly well-profiled, highly aware cells. How does it actually draw the borders?
Right, good catch. To get the actual neighborhoods, this data passes through a fully connected linear layer that applies a softmax function.
Whenever I hear softmax in AI, I think of probabilities. Is it hedging its bets? Like, instead of saying you are in neighborhood A, end of story, it's saying like, I'm 60% sure this is neighborhood A.
Precisely. It gives us a soft TCN assignment, a probability matrix. It doesn't draw a hard border yet. A cell might be 60% likely to be in neighborhood A and 40% in neighborhood B.
Okay, but we need hard borders eventually.
We do. And to force the AI to make a confident decision, CytoCommunity uses a really brilliant mathematical trick called a min-cut-based loss function.
Min-cut, minimum cut. Are we like taking a pair of scissors to our spider web?
Exactly. Imagine a pair of scissors. The goal of the min-cut scissors is to cut the vast web into equal-sized island neighborhoods, but it wants to snip the absolute fewest number of threads possible.
Oh, wow.
Yeah, the math literally penalizes the algorithm for cutting too many connections.
That makes total sense biologically. Because cells in the same functional neighborhood are going to be tightly packed and highly connected, right? So the scissors naturally find the fault lines where the connections are already sparse.
You nailed it. And the math also pushes the probabilities to be orthogonal.
Meaning?
In plain English, it forces the AI off the fence. It hates 50-50 probabilities. It pushes the AI to eventually make an unambiguous, hard decision about which neighborhood a cell belongs to.
But, what if it makes a mistake? What if the math just has a weird fluke on one of the cuts?
That is where ensemble learning comes in. The wisdom of the crowd.
Oh, I love ensemble learning.
Right. CytoCommunity doesn't just run the scissor process once. It runs multiple models simultaneously, usually about 20 different models. Each model explores slightly different probabilities.
Okay.
Then the algorithm looks at all 20 models and takes a simple majority vote to lock in the final hard boundaries.
Okay, let's pause and synthesize this engine because it is just elegant. So, bullet recap for you listening. The AI casts a KNN spiderweb over the physical cells. It tags every cell with a clear one-hot math barcode. It filters the signals through a graph neural network with RealU to calculate probabilities using softmax.
Perfect.
Then it uses min-cut mathematical scissors to cut the web into islands by snipping the fewest threads possible. And finally, it runs that whole process 20 times and takes a vote to lock in the absolute best map.
That's exactly how it works.
So an interactive prompt for you listening. If you were designing an algorithm to dictate your own medical treatment, would you rather trust one single brilliant calculation or the majority vote of 20 slightly different rigorous calculations?
I know which one I'd pick.
Right. That consensus approach makes this tool incredibly stable.
And that stability is vital because everything we just discussed, that's just the baseline.
Wait, really?
Yeah, that is unsupervised learning. It relies purely on the min-cut loss function to map individual unlabeled tissue maps. Finding hidden borders in one single piece of tissue is great, but in the real world of medicine, mapping one patient just isn't enough.
Right. It's fascinating to map one single tumor, but the real holy grail is, like comparing 50 cancer patients who survived with 50 patients who sadly didn't, to see which specific cellular neighborhoods predict survival. How does it jump from a single map to a huge population study?
For that, we shift to the supervised superpower of the algorithm. CytoCommunity introduces a weakly supervised graph partitioning problem, using what's called a differentiable graph pooling layer.
Graph pooling. Let me bring back our map analogy here. If the original spiderweb graph is like Street View, where we're mapping individual cells one by one as graph pooling, basically zooming out to city view, where an entire massive neighborhood becomes just a single dot on the map, and the lines between dots represent how whole neighborhoods interact.
That is exactly what happens. The pooled nodes are mathematically averaged into what's called a whole graph embedding vector. It's a high-level summary of the entire tissue slide.
Okay.
And because this is supervised, we now feed the AI the actual patient labels. We tell it, this was a high-risk patient or this was a low-risk patient.
So the AI is essentially trying to multitask.
Yeah.
It's trying to draw the neighborhood borders while simultaneously trying to guess if the patient lived or died based on those borders.
Multitasking is the perfect word. It uses a dual loss function. It combines the min-cut scissor math, which focuses on partitioning and drawing good borders, with cross-entropy loss, which is a classic formula for sample classification, or guessing the patient's label. It plays this mathematical tug-of-war between the two.
How does it balance that tug-of-war, though? If it focuses too much on guessing the clinical outcome, couldn't it draw fake, biologically impossible neighborhoods just to make the math work?
Exactly, which is why the balance is strictly weighted. The system uses a parameter called beta, and beta is usually set to 0.9.
Okay, what does that mean in practice?
It means it dedicates 90% of its effort to the min-cut partition, ensuring the biology is real and the borders make physical sense. Only 10%, the 1 minus beta, is focused on guessing the clinical outcome.
Oh, I see.
Yeah, this subtle pressure ensures the neighborhoods are biologically realistic, but optimized just enough to highlight the spatial differences that actually dictate patient survival.
Okay, let's do another mini recap here on this perspective shift. Unsupervised learning gives us the street view of a single map. Supervised learning uses graph pooling to zoom out to the city view summary. And by balancing the math with that beta of 0.9, the AI connects specific tissue layouts directly to patient outcomes. Just imagine the power of that. You feed a supercomputer 100 tumor slides, and it automatically highlights the exact cellular neighborhood, the specific biological zip code that causes a patient to relapse.
Which brings us to the actual moment of truth, because elegant math is just theory until you test it on real tissue, right? The researchers actually unleashed CytoCommunity on complex biological data sets, and the level of evidence they found was just staggering.
Let's get into the case studies. I know they started with healthy tissue. They tested it on the spleen and the brain, right? Why pick those two?
Because they are notoriously difficult to map. They looked at a mouse spleen using CODEX technology, which measures 30 different proteins. The spleen contains these elusive, really hard to define structures called marginal zones.
And how did the older tools do with that?
Oh, tools like UTAG and spatial LDA completely failed. They either missed them entirely or drew incredibly messy, blurred boundaries. But CytoCommunity mapped them with stunning precision. It achieved the highest macro F1 and AMI scores, meaning its borders matched the biological ground truth established by expert pathologists almost perfectly.
Wow. And the brain.
For the brain, they looked at the mouse hypothalamus using MERFISH technology, tracking 155 different genes.
Okay, that's a lot more data.
Yeah, and the hypothalamus has this incredibly complex neuroanatomy with symmetric brain nuclei.
Symmetric meaning like mirror images.
Exactly. Structures with names like BNST, MPA, MPN, and VLPO. They are essentially perfect mirror images of cell communities on both sides of the brain.
I can imagine that's tricky for an AI.
Older spatial transcriptomics tools just created a blurry mess when trying to map them. But CytoCommunity successfully mapped the exact mirror boundaries, matching the complex neuroanatomy gold standards better than all existing tools.
Okay, so it handles the intricate architecture of healthy tissue flawlessly. But let's get into the high-stakes arena, cancer. What happens when this AI looks at tumors? Let's talk about colorectal cancer, or CRC.
Right. So in CRC, they used supervised learning to compare the tissue maps of low-risk patients who generally have better survival rates, obviously against high-risk patients.
Okay.
In the low-risk patients, CytoCommunity discovered a very specific neighborhood, which they labeled TCN1. And this neighborhood was heavily enriched with B cells.
B cells are immune cells, right? So this neighborhood is basically like a base camp for the immune system.
Exactly. They're called tertiary lymphoid structures. It's the body literally setting up a localized forward operating base right next to the tumor to fight it. But in the high-risk patients, that B-cell base camp was completely missing.
Oh, wow. What replaced it?
A different neighborhood, TCN2, which was packed with granulocytes and neutrophils. And in this specific context, those cells actually promote tumor growth. But here's where CytoCommunity showed its true power. It didn't just find isolated neighborhoods. It actually found active cell communication between them.
Wait, the neighborhoods were talking to each other?
Yes. In the low-risk patients, the AI revealed that the granulocytes in TCN2 were spatially interacting very closely with specialized double-positive macrophages in another neighborhood, TCN6.
Okay, what does that interaction do?
This physical proximity allowed them to collaborate and exert a massive anti-tumor effect. The AI mapped the exact physical communication lines between the communities to fight the tumor.
So CytoCommunity isn't just like a cartographer drawing lines on a map. It's a strategist mapping out the supply lines and communication networks on a battlefield.
That is the perfect way to frame it. And we see an even darker, more aggressive version of this battlefield in breast cancer.
Let's hear it.
They analyzed breast cancer tissues using IMC and MEBI-TOF technologies. And for the high-risk patients, the ones with the worst clinical outcomes, CytoCommunity consistently identified a lethal, highly specific neighborhood. They called it TCN4.
What makes TCN4 so lethal? What is actually inside that neighborhood?
It is heavily populated with cancer-associated fibroblasts, or CFs, and endothelial cells.
Okay, what's the biological mechanism there?
The mechanism is really sinister. When a tumor grows very aggressively, it outgrows its own blood supply. This creates hypoxia, a severe lack of oxygen in that area.
You would think a lack of oxygen would just kill the tumor.
Right. You'd think so, but it actually just kills the weak tumor cells, leaving only the most aggressive mutant clones to survive and expand. But worse, that hypoxic environment corrupts the normal, healthy fibroblasts living in the tissue. It transforms them into these CFs.
So the tumor is essentially radicalizing the local citizens to work for it.
Yes, and those radicalized CHAFs in neighborhood TCN4 go straight to work. They promote angiogenesis, meaning they force the endothelial cells to rapidly build new blood vessels to feed the starving tumor.
Unbelievable.
And simultaneously, this dense neighborhood acts as a physical wall. It's an exclusion barrier that physically blocks your immune cells from entering the tumor bed to fight it.
That is chilling. The AI found the exact physical neighborhood where the tumor builds its fortress, grows its own food supply, and locks the immune system out.
And it did it automatically. This completely revolutionizes how we look at cancer, because purely pathology-based subtyping, just looking at the shape of the tumor cells themselves, it often misses these vast, subtle spatial relationships in the environment around the tumor.
Let's do our final major recap here, because the clinical implications are huge. CytoCommunity proved its accuracy by finding hidden marginal zones in the healthy spleen and symmetric nuclei in the brain.
Yep.
In colorectal cancer, it identified protective B-cell base camps in low-risk survivors and tumor-promoting granulocyte neighborhoods in high-risk patients. And in breast cancer, it mapped out TCN4, this physical fortress, where corrupted CCAFs build blood vessel networks to feed the tumor and block the immune system.
Spot on.
So I really want you listening to ponder this. If we know exactly which physical neighborhood is supplying the tumor, could the next generation of cancer drugs be designed to target the zoning laws of the tissue rather than just blindly attacking the tumor cell itself?
That is really the ultimate goal of the next decade of medicine, to disrupt the microenvironment. You don't just bomb the fortress, you cut off the supply lines.
So synthesizing this entire journey, we started in the messy, chaotic world of raw gene expression, where older tools were just getting lost in the noise. We walked through the elegant mathematical engine of CytoCommunity, casting physical KNN spiderwebs over the cells, tagging them with one-hot math par codes, filtering them through neural networks, and using min-cut scissors to draw precise biological borders.
Right.
We zoomed out to the city view, using graph pooling to compare outcomes across dozens of patients. And we arrived at these life-saving clinical insights, uncovering the hidden communication networks and fortresses inside colorectal and breast cancers.
It is a monumental step. But, as with all cutting-edge science, if we connect this to the broader reality, we do have to acknowledge a current limitation.
Okay, what's the catch?
The algorithm requires pre-annotated cell types to function. You have to already know the identity of the cells before you can map their neighborhoods.
So for lower resolution spatial mapping, where one single spot on a slide might contain a microscopic mix of 3 or 4 different cells just smushed together, you can't just plug this AI in and hit play.
Correct. For lower resolution data, researchers must first use separate mathematical deconvolution methods to untangle and guess the cell types in that spot before they can feed it to CytoCommunity.
Makes sense.
But looking forward, the future of this technology actually points toward analyzing spatiotemporal omics.
Spatiotemporal, so adding the fourth dimension, time.
Exactly. Not just taking a static snapshot, but watching these neighborhoods evolve, shift, and communicate over time as a disease aggressively progresses or, as a patient undergoes chemotherapy.
Which brings us perfectly back to our opening city map. Think about the city or town you live in right now. Neighborhoods aren't static. Over the years, they gentrify, they decay, they expand, new highways cut right through them. Our bodies are exactly the same dynamic landscapes.
They absolutely are.
So I want to leave you with a final provocative thought to mull over. If algorithms like CytoCommunity can now look at the physical spatial arrangement of our cells and predict our medical future based purely on the neighborhoods those cells live in, what does this mean for the absolute peak of personalized medicine? Could we soon have digital twins of our own, unique tissue neighborhoods living inside a hospital supercomputer?
Now that is a wild thought.
Just imagine your doctor running thousands of digital simulations on your specific tumor map, testing 100 different therapies to see exactly which drug will disrupt the bad neighborhoods, collapse the tumor's fortress, and leave your healthy neighborhoods perfectly intact. It's a completely new paradigm for healing, something to really think about the next time you walk through your own city.